At the end of the last chapter, Earth had finally done something no world we know of had managed before. It had grown an ocean, vast, connected, chemically restless, and stable enough to last. It was not yet a hospitable place. Volcanoes still burned along its edges. The largest impacts had not entirely stopped. The sky above it was still without a breath of oxygen. But somewhere in that ocean, sometime between roughly 3.8 and 3.5 billion years ago, something happened that has never stopped happening since. A collection of chemistry, carbon, hydrogen, oxygen, nitrogen, phosphorus, sulfur, arranged in the right way, in the right place, crossed a line that, as far as we have ever been able to tell, nothing had crossed before. It began to do something that looked unmistakably like being alive. This is the story of that crossing, and of how honestly uncertain and how genuinely fascinating our best current answers for it really are. Before we go further, it is worth being completely honest about something. No scientist currently knows exactly how life began on Earth. This is not a gap waiting to be filled by simply reading more research. The origin of life, a field researchers call abiogenesis, remains one of the most actively studied and most genuinely unresolved questions in all of science. What we have instead of a single confirmed answer is something arguably more interesting. Several serious, evidence-based competing hypotheses, each grounded in real chemistry, real geology, and real experiments, each attempting to explain how non-living matter could have organized itself into something capable of growing, reacting to its environment, and copying itself. What follows in this episode is not a settled account. It is a tour through the leading ideas science currently takes seriously, and a look at what each one gets right, and where each one still runs into genuine, unresolved difficulty. The oldest of these modern scientific hypotheses dates back to the 1920s, when biochemist Alexander Oparin in the Soviet Union and geneticist JBS Haldane in Britain independently proposed a similar idea, now often called the primordial soup hypothesis. Their proposal was elegant in its simplicity, given an early atmosphere rich in simple gases and a source of energy, lightning, volcanic heat, or ultraviolet light from the sun. Basic organic molecules, the building blocks of life, could plausibly assemble themselves in the ocean, given enough time. In 1953, graduate student Stanley Miller and his advisor Harold Urey put this idea to a direct experimental test. They built a sealed glass apparatus containing water, methane, ammonia, and hydrogen, gases they believed represented the early atmosphere, and repeatedly sparked the mixture with electricity, simulating lightning. Within just a week, the water inside had turned a distinct, telltale pink-brown. Analysis revealed that a startling variety of amino acids, the fundamental building blocks of proteins, had spontaneously formed, along with several other biologically relevant organic compounds. It remains one of the most famous single experiments in the history of chemistry. There is, however, a significant complication, and in the spirit of scientific honesty, it is worth stating plainly. As we discussed in earlier episodes of this series, more recent geochemical modeling suggests Earth's actual early atmosphere was probably dominated by carbon dioxide and nitrogen, rather than the strongly hydrogen-rich methane and ammonia mixture Miller and Urey used. Under a more realistic, less chemically reducing atmosphere, similar spark discharge experiments tend to produce meaningfully smaller quantities of amino acids. This does not eliminate the primordial soup hypothesis outright. Some organic synthesis under these more realistic conditions still occurs, and some researchers argue certain localized environments, such as sites near volcanic outgassing, could still have been more hydrogen-rich than the average early atmosphere. But it has meaningfully shifted scientific attention toward looking for environments that could supply their own concentrated chemical energy directly, rather than relying primarily on the atmosphere and lightning. That search for a more chemically active setting leads us to the deep ocean floor, and to one of the most compelling alternative hypotheses currently studied, that life began not at the surface, but at hydrothermal vents. Modern hydrothermal vents come in more than one variety, and the distinction matters. Famous black smoker vents, first discovered in 1977, spew acidic, superheated fluid, often exceeding 300 degrees Celsius. dramatic but arguably too hot and too acidic to easily assemble fragile early biological molecules. But a different kind of vent system, cooler, alkaline, and driven by chemical reactions between seawater and mantle rock, rather than direct volcanic heat, has become the focus of a specific and influential origin of life hypothesis. A real modern example of such a system, called the Lost City hydrothermal field, was discovered in the year 2000 in the mid-Atlantic. Its vents release warm, alkaline fluid and are built from porous carbonate rock riddled with countless microscopic mineral chambers. The core insight of this hypothesis is genuinely elegant. These porous mineral structures naturally create a chemical gradient, a difference in acidity between the alkaline fluid inside the vent and the more acidic ocean water outside it. Every living cell on Earth today, without exception, generates the energy it needs by maintaining a very similar kind of gradient, called a proton gradient, across a membrane, using it to power a remarkable molecular machine called ATP synthesis. Proponents of this hypothesis argue that this is unlikely to be a coincidence. If early chemical precursors to life first organized themselves within the microscopic mineral pores of a vent like this, naturally bathed in a steady, ready-made proton gradient, they would have had access to a continuous, concentrated source of usable energy, potentially allowing the earliest steps of what we would call metabolism to begin before life had even evolved a membrane or a genetic code of its own. A third serious hypothesis, notably, brings us back toward dry land, and toward a location relevant to a piece of evidence we'll return to later in this very episode. Some researchers argue that deep-sea vents, despite their appealing chemistry, have a significant drawback. They are permanently underwater in a stable, unchanging environment. Certain critical chemical steps in building the complex molecules of life, however, appear in laboratory experiments to require repeated cycles of wetting and drying, conditions much more easily found in shallow land-based hot springs that periodically evaporate, rather than in a permanently submerged deep-sea vent. These wet-dry cycles are thought to be particularly good at encouraging simple organic molecules to link together into longer chains, and at helping fatty, oily molecules called lipids spontaneously assemble into small, hollow spheres called vesicles, simple, cell-like membrane compartments that many researchers consider a plausible ancestor of the first true cell membrane. Intriguingly, some of the best geological evidence supporting this specific land-based hypothesis comes from a very old, very well-studied piece of Australian geology called the Dresser Formation in the Pilbara region of Western Australia, dated to approximately 3.48 billion years old. Researchers examining this formation in detail have identified mineral textures consistent with ancient, land-based hot spring pools rather than a deep marine setting. We are not yet finished with the Pilbara. As we'll see shortly, this same remarkable stretch of ancient Australian rock holds still more significance for this story. Underneath all three of these environmental hypotheses sits a deeper, more fundamental scientific puzzle. Which came first, a self-copying genetic molecule or a self-sustaining chemical metabolism? Modern cells rely on an intricate partnership between DNA, which stores genetic information, and proteins, which carry out nearly all of a cell's actual chemical work. The trouble for anyone trying to explain the origin of this system is that DNA generally requires proteins to copy itself accurately, while proteins are generally built according to the instructions stored in DNA. Neither seems capable of easily appearing without the other already being in place, a genuine chicken and egg problem. One influential proposed solution, generally called the RNA world hypothesis, points to a close chemical cousin of DNA called RNA. Unlike DNA, RNA is capable of folding into complex three-dimensional shapes, and in the 1980s, biochemists Thomas Cech and Sidney Altman discovered that certain RNA molecules, called ribozymes, can actually catalyze chemical reactions on their own, functioning much like simple protein-based enzymes. This raised a genuinely exciting possibility that RNA alone in the very earliest stages of life might have been capable of doing the job of both DNA and protein simultaneously, storing basic genetic information while also catalyzing its own replication and other useful chemical reactions. Before the more specialized, more efficient DNA and protein system we see in every modern cell gradually evolved around it. The RNA world hypothesis remains genuinely popular among origin of life researchers, though it too faces real unresolved challenges. particularly around how the first RNA molecules could have reliably formed and accumulated under plausible early Earth conditions in the first place. Whatever combination of these processes actually occurred, they eventually converged. At some point on a single organism whose descendants are still with us today, in every corner of this planet, an organism scientists call LUCA, the last universal common ancestor. It is important to be precise about what this term actually means. LUCA was almost certainly not the very first living thing to exist. It was, more specifically, the most recent single population of organisms from which every form of life alive today, every bacterium, every archaeon, every plant, fungus, and animal, including us, is descended. We have never found a fossil of LUCA, and given how fragile and microscopic any such organism would have been, we likely never will. Instead, scientists reconstruct what LUCA was probably like using comparative genomics. By carefully comparing the genes shared across the most deeply diverged branches of the tree of life, bacteria and archaea, researchers can infer which genes and which biological functions were probably already present in their last shared ancestor. It's worth pausing on how remarkable this method actually is. Nobody has traveled back in time. Instead, by comparing the genetic spelling of thousands of genes across thousands of modern species and working out which shared features are too consistent and too fundamental to have evolved independently more than once, researchers can effectively work backward through billions of years of evolution to sketch a biochemical portrait of an organism nobody has ever seen. One especially detailed reconstruction effort, published in 2016, concluded that LUCA was very likely a single-celled organism that thrived in extremely hot water, entirely without oxygen. Obtaining its energy by chemically combining hydrogen gas with carbon dioxide, a metabolic strategy still used by certain microbes living around hydrothermal vents today. That reconstructed profile, heat-loving, oxygen-free, chemically powered by hydrogen and carbon dioxide, lines up remarkably closely with the conditions proposed by the alkaline hydrothermal vent hypothesis we discussed earlier, which many researchers consider a meaningful, mutually reinforcing piece of evidence, even though it does not resolve the debate entirely. As for exactly when LUCA existed, that too remains genuinely uncertain. Molecular clock studies, which estimate evolutionary timing based on the accumulated rate of genetic change, have produced a range of estimates. One detailed 2018 analysis placed LUCA's most likely age at somewhere in the range of 3.9 to 4.5 billion years ago. Though the method carries substantial uncertainty, and other researchers favor a somewhat younger date, closer to 3.5 to 3.8 billion years. Whatever LUCA's exact birthday, we do have direct, physical, geological evidence that life existed on Earth by roughly 3.5 billion years ago. And once again, that evidence comes from the ancient rock of Western Australia's Pilbara region. The evidence takes the form of structures called stromatolites, layered, dome-shaped or column-shaped rock formations built up, quite literally one thin layer at a time, by colonies of microorganisms living in shallow water. As these microbial mats grew, they trapped fine sediment and, in some cases, encouraged the precipitation of minerals directly from the surrounding water, gradually constructing a durable, layered rock structure as a kind of accidental architectural byproduct of simply being alive. It's worth being clear here. These earliest stromatolites were almost certainly not built by cyanobacteria, the oxygen-producing microbes responsible for the Great Oxidation Event. We'll cover 2 episodes from now. Free oxygen would not begin meaningfully accumulating in Earth's atmosphere for another billion years. These early mat-building microbes were most likely simpler anoxygenic organisms, obtaining energy through chemistry that did not release oxygen as a byproduct. The best-known and most rigorously documented example comes from the Strelley Pool Formation, also in the Pilbara, where a research team led by geobiologist Abigail Allwood documented an extensive, exceptionally well-preserved reef-like network of stromatolites, precisely dated to approximately 3.43 billion years old. Critically, their detailed analysis of the structure's shape, internal layering, and spatial distribution argued strongly against these formations being simple, non-biological mineral accidents and in favor of a genuinely biological origin. Nearby, in the same broader Pilbara region, the Dresser Formation, the same rock formation we mentioned earlier in connection with the land-based hot spring hypothesis, has also produced stromatolite-like structures, Along with other chemical and textural evidence, some researchers interpret as consistent with early microbial activity, dated to approximately 3.48 billion years old. It is worth briefly mentioning that Australia does not hold the only claim to extremely ancient life. In 2016, a research team reported what they interpreted as even older stromatolite-like structures, dated to roughly 3.7 billion years old, from the Isua Greenstone Belt in Greenland. That claim, however, was seriously challenged just 2 years later by a separate research team, including some of the same Australian stromatolite specialists, who re-examined the Greenland site and argued the structures were more likely the product of purely geological deformation, with no clear evidence of biological origin. This kind of back and forth, played out publicly in one of the world's leading scientific journals, is not a weakness in the scientific process. It is the process working exactly as intended. Extraordinary claims about the deep history of life are, appropriately, held to an extraordinarily high standard of scrutiny. And the Australian stromatolite evidence has, so far, proven considerably more durable under that scrutiny than its Greenland rival. Let us take stock of this remarkable, still unfinished chapter. Sometime between roughly 3.8 and 3.5 billion years ago, in an ocean we described forming just one episode ago, simple chemistry crossed into biology. We do not know with certainty exactly where this happened, whether in the sunless dark of an alkaline hydrothermal vent, in the wet-dry cycling of a mineral-rich hot spring pool on land, or in some combination or entirely different setting we haven't yet fully considered. We do not know with certainty whether genetics or metabolism came first, though the RNA world hypothesis remains a serious leading candidate for bridging that gap. What we do know is that all of it, however it happened, eventually converged into a single ancestral population, LUCA, a heat-loving, oxygen-free organism whose direct descendants would go on to become every living thing that has ever existed on this planet, including eventually you. And we know, from durable, repeatedly tested physical evidence preserved in the ancient rock of Western Australia, that living, mat-building microbial communities were unambiguously, demonstrably present and thriving by at least 3.43 billion years ago, meaning life in some form had very likely already existed for hundreds of millions of years before that, quietly building the oldest confirmed biological structures we have ever found. Earth, once a lifeless ball of accreting dust, then a molten, bombarded wasteland, then a poison, rain-soaked ocean world had finally genuinely become something new, a living planet. It would remain almost entirely microscopic, almost entirely invisible, for a staggeringly long time yet to come. But the line, once crossed, was never uncrossed again. That is where we go next. See you in the next video.